Now, Shannon has also optimized the imaging technology. Initially, this involved looking through a microscope for prolonged periods of time and making a subjective call of what is a CTC and what isn't. This is a complex platform using a BioView type of semi-automated imaging device, which really will scan the entire platform of the chips, use a number of integrated measures, including the signal intensity, the actually location in three-dimensional space, because this is still a chamber where the center of fluorescence is, and then a number of other parameters for cell shape and size that really make sure that what we call CTCs are true CTCs. And you can, again, image the entire chip. It turns out, depending on the levels of EpCAM expression in the cells, you either capture most of the cells towards the inlet, and then there's a decay pattern, or you capture them more towards the middle of the chip, again, depending on the levels of antigens in the cells. And this has really made it possible to set particular thresholds and parameters that would enable clinical trials of this technology. Now, let me touch on a couple applications of the CTC type approaches. And I want to first talk about metastatic cancer. Again, the idea of using this not just for numbers of CTCs, but to identify molecular markers that are very relevant. A little bit of background. We've been obsessed with lung cancer. It turns out that about 6 years ago, our group and a group at the Broad Institute defined a genetically defined type of lung cancer, which you can see here, which is rare. It's about 10% of lung cancer in the US. It tends to arise in non-smokers, so it seems not to be triggered by cigarette toxins, and the mutational profile is quite different from those that are induced by cigarette intake. The histology tends to be primarily adenocarcinomas, often with some bronchoalveolar differentiation. And for reasons that are still fascinating and completely not understood, it's about twice more common, twice more prevalent in women than in men, and in patients or in individuals of Asian ethnic background than in Caucasians. It's a somatic mutation, so we don't really understand this genetic susceptibility or environmental susceptibility, but quite remarkable. What's clearly evident is that if you treat patients with EGF receptor inhibitors, gefitinib or erlotinib, if their tumor has this mutation, you see dramatic responses, whereas in patients who don't have these mutations, the responses are really, really quite modest. And we've heard yesterday about the whole mechanism of oncogene addiction. In this particular case, there are two particular mechanisms that have been implicated. The first is that the mutant receptor binds to ATP less well than the wild-type receptor, which makes it easier for these drugs, which compete with ATP for binding, to suppress binding of ATP to the enzymatic pocket. That turns out to be quite relevant because at the therapeutic achievable doses, that's exactly where there's a differential between the mutant receptor and the wild-type receptor. So you can achieve complete inhibition of mutant receptor signaling at therapeutic doses, which are more borderline for the wild-type receptor. Probably the most important mechanism is that mutant receptors signal differently. This has been most well defined by the work of Jeff Engelman and Lou Cantley, which suggests that the mutant receptors signal through ERBB3 primarily to drive survival signals through AKT. And what happens then is if you withdraw these signals from mutant EGFR-driven cells, the cells undergo apoptosis. If you withdraw the same signals from cells with wild-type EGF receptor, they don't have the same impact. So this is by way of genetic background. In terms of detection, we're somewhat lucky in that most of these mutations are specific. So there are two types of mutations. You see these deletions mutations here, these specific mutations here, which together account for about 80 to 85% of the activating EGFR mutations which drive these cancers. And when the cancers become resistant, which happens after about 6 months to a year, there's a common recurrent gatekeeper mutation that suppresses drug binding, which again is present in about 50% of cases. So there are known mutations that we can look for, and what we're able to do is look at patients with known EGFR mutant cancers, look at their CTCs, mostly by allele-specific PCR, in some cases by direct sequencing. And what we found is that, yes, indeed, we can identify the mutation in the CTCs. In 11 out of 12 cases, we're correct. We can find the emergence of the drug resistance mutation. It's more common during the course of treatment. And in fact, we find that those in whom we don't see the mutation at the time of initiation of treatment have a longer response than those in whom you can find a very, very rare resistance mutation, which then quickly becomes selected for during the course of therapy. And then, to our great surprise, we find, particularly in patients who had a dramatic response and when the tumor grows back a year later, the types of mutations that we find are sometimes different than we could identify a year ago at the time of initial diagnosis. So it really points to the fact that you need to identify and follow these mutations during the course of therapy. Now, we can do this. This is actually a number of cases we could follow for about a year, where we can see in green the CAT scan results showing initial responses and then recurrence of the tumor. And in red, we can see decreases in CTC numbers, increasing CTC numbers at the time of relapse. And at every point along this curve, we can genotype the tumor and see what is the dominant genetic makeup of the tumor. And the more effective the treatment is, the more effective the treatment itself has an impact on the genetic composition of the tumor. Now, do we care about this? This is a paper that was published a year ago now in the New England Journal from an Asian group led by Tony Mock, showing that yes, if you identify non-smokers with lung cancer and you give them either chemotherapy or an EGF inhibitor, there are all kinds of different results that you get. If you give them chemotherapy, they all do about the same. If they have an EGF receptor mutation and you block their EGFR with an inhibitor, they do very well. If they don't have an EGF receptor mutation and you give them an EGF receptor inhibitor, they do very badly. And that really suggests that there actually are a number of different genetic mechanisms for non-smokers' lung cancer. This is a paper that was published a few years ago by Dr. Soba and Dr. Mano's group, identifying yet another genetic abnormality, a translocation involving the ALK kinase in lung cancer. We were fortunate in that we were able to set up a number of facilities at MGH. This is one that was led by Jeff Settleman, looking at a whole panel of cancer cell lines, treating them with an ALK inhibitor, and finding that, yes, indeed, ALK inhibitors are specifically toxic, specifically lethal to tumors that have ALK mutations, be they neuroblastomas, lymphomas, or lung cancers. And we went from that to a pathology collaboration, identifying, as we now do routinely, all kinds of genetic abnormalities in all patients with lung cancer who are treated at Mass General, and identifying subsets with different types of mutations who could be enrolled in real time in our clinical trials. And it's particularly important because what we typically do very well is we treat patients with drugs, and then we go back, collect the tumor, and try to figure out who had what mutation. To be able to do this on day one, at the time when the patient presents, means that you can then identify patients with ALK mutations and then treat them with an ALK inhibitor if appropriate. That's precisely what was done, and this is a trial which is just completed now, led by Eunice Kwok, John Iafredi, Jeff Clark, and Alice Shaw, in which they typed 600 patients. This is an international trial which was led by MGH and involved testing 600 lung cancer patients for ALK mutations, identifying 30 with these mutations, and then enrolling about 18 of these patients. This is an early result, 18 patients on this trial with a response rate of about 85%. So this is really the idea of identifying patients up front with a genetic marker, if they have that marker, even if it's a rare marker in the overall population, using targeted therapies against tumors with this particular marker. The responses are very dramatic, just like EGF receptor inhibitor. You can see melting away of tumors within 2 cycles of this ALK inhibitor. And when we start looking at CTCs, you can see the same thing. Within 2 weeks of treating patients with ALK inhibitors, you see a drop in the number of CTCs. And we're now trying to, you can see staining for CTCs with this ALK inhibitor from cell signaling technologies. And you can see now that we're trying to study whether we can identify both mutations as well as responses using CTCs. So to summarize this lung cancer part, what I would say is that lung cancer has evolved now in terms of a modeled cancer, which is quite common in the population but has specifically defined subclasses. And there's at least 2 that are common in non-smokers with lung cancer: EGF receptor mutations, ALK rearrangements. And you can no longer guess which treatment you should give to which patient. You actually have to identify which genetic lesion is present in which cancer. You need to know it at the time of initial treatment, and you need to follow patients during treatment, because we're now studying the mechanisms of acquired resistance to these drugs. And there are very different mechanisms of resistance that are acquired, and each of these different mechanisms will have a separate drug that may be effective. So again, we're looking at a very different phase in the treatment of cancer with targeted therapies, and knowing what you're treating becomes absolutely critical.